So just while everybody is coming in, just hello, welcome to Imaging One World. And most people, I think, have been here before. For those who haven't, we have this lecture every Monday from 1 to 2. It started in lockdown as an idea to keep the imaging community going, and turned into quite a successful, continuous, weekly event. And we initially, I think, were quite hesitant to invite people from very far abroad because of the time difference, but then it didn't seem to deter people, and we had quite wonderful, eclectic lecture kind of community from all over the world, really. Last week, last speaker we thought was joining us from Colombia, but she had already moved to China. So we really are an international lecture series. I'm very proud today to have Carsten Marr joining us from Germany, from Munich, where he has been for many steps in his career. He's a world famous person and scientist for bioinformatics, AI applications to health questions, and currently, and for quite a few years, he's the director of the Institute of AI for Health, AIH, at the Helmholtz Zentrum in Munich. And yeah, he received many awards, the Erwin Schrödinger Prize and CSP2 Prize in Systems Biology. He was in Edinburgh, many other places. He's been, I'm sure, a visiting scientist, but his group is now focused and based in Munich, the Helmholtz Zentrum, and he's talking about artificial intelligence and computational hematopathology. And please, Carsten, enlighten us, tell us about your research and your interests and what the community does and your group.
Thanks, Stephanie. Thanks for the invitation and the nice introduction. I was really happy to be able to speak in that series, which is really interesting collection of speakers. I'm very happy to show you our recent works here and also get your feedback on that. So let me start by introducing the lab, which is basically divided into the one side, which is rather deep learning focused and trying to boost image-based diagnostics. So we have a translational AI focus here. And on the other side, we develop mechanistic models to predict single-cell kinetics. Also with a clinical focus, so we try to take data from patients, some measurements from the clinics, and understand what cells or tissues are doing. And these are basically two things. And of course, today, since this UK imaging series, of course, I'm going to focus on the left-hand part, but I'm happy to talk about the right-hand part whenever asked. So why do we look into biomedical and clinical data? Well, there's a data explosion expected in the healthcare series, actually, in that white paper here, where people looked into 5 different sectors. The data the growth rate of the data is expected to be larger for healthcare than for any of the others, so including manufacturing, finance services, and media. And why is that? Well, because I guess on the one hand side, we carry the variables with us, many of us, right, all the time, collecting our blood pressure or temperature or health status as much as we can. On the other hand, the clinics and the doctors, the practitioners are getting our data in more and more in a clean, organized, digital fashion, allowing us to use and exploit that kind of data. So within the next couple of years, we will have more and more of patient clinical data at our hands, which will allow us to look into connections between different measurements, connections between observables in people, but also in societies. And of course, we just really need methods that allow us to deal with that kind of data. And our part is to look into, well, blood, for example. That's actually one of the foci of my labs, also since I have the ERC on that. And when looking into blood, it's good to look at single cells, because the blood cells can be nicely isolated and then analyzed. And if you look into single cells, you might first wonder how many human cells do we consist of? And this is an amazing number, 30 billion human cells. It's a rough estimate of each one of us, right? So we are basically human, a bag of human cells. Fun fact, we probably harbor the same number of non-human cells in our gut. So you could also look at us as something like a bag of human cells carrying another bag of bacterial cells in us. Anyways, let's go back to blood. Most of these human cells actually are blood cells. 25 billion are blood cells, and most of those guys actually are the red blood cells in our blood, right? They carry oxygen, and they don't have a nucleus, they're pretty tiny. So most of our cells are blood cells. And then that is an amazing number, right? But maybe even more amazing is the fact that we produce 500 billion every day. Did I say billion? Well, it's actually trillion. Depending on which system you use. I'm always getting confused. In Germany we have Millionen, Milliarde, Billionen, Billiarde. So Stephanie, maybe you
lose
can...
kind of millions of cells as well every day, no? We kind of
exactly.
lose,
so we
lose them actually over
roughly the same amount, right? Because otherwise we would grow. Right, but we produce newly functional cells every day, and this is now, sorry if I said it wrong. So this is 500 billion, right? 10 to the 6, 9, 11. So that's the number of cells we produce. And the question was, how do we do that? And we look into that from different perspectives. And interestingly, people looked at that for hundreds of years because you could analyze blood pretty easily, right? You could just take a drop of blood and look into it, for example, under microscopes. Once you had one, that's why blood is probably the best tissue of all. And we have understood quite nicely how blood cells are produced. And the idea is that we have blood stem cells in our bone marrow sitting there, and then differentiating via this hierarchy of states that you see here, from the state at the apex, the HSC, hemoglobin stem cell state, over a couple of progenitor cells, into the mature functional states at the bottom, right? So platelets, erythrocytes, granulocytes, macrophages, all these cells have different tasks, and they all stem from the same stem cells in our bone marrow, which is very interesting. Now, we understand a lot about that, and together with my group, actually a couple of years back, when the group was consisting mainly of me and 2 PhDs, we looked if we can use machine learning for predicting the fate of a blood stem cell. And the task was relatively simple. Together with our colleague Tim Schröder at that time, Tim took photos, time-lapse movies of differentiating mouse blood stem cells. And at some point, there was a decision made of these blood stem cells going either into the one direction or the other, right? And the question was, can we discriminate megakaryocyte erythrocyte progenitors from the other type, granulocyte monocyte progenitors, just by looking at the images of those cells? And if you look at these images, you might say, might look a bit different, but actually they look pretty similar. So we tried to figure out if a machine learning model can do that. And we were one of the first people, I guess, in biology applying a deep neural network to a biological question. And we could show that actually we can discriminate that when training a deep neural network. We can discriminate the MEPs on the left-hand side from the GMPs on the right-hand side. And we can do that in particular for cells that turn a marker on, a fluorescent marker, which is basically the ground truth, right? So we know which is which. But interestingly enough, we can do that also before the marker turns on. So basically, we can use the morphology of these cells to predict what they will become in terms of upregulation of the marker expression. We can do that, or we could do that at that time, right? 36 hours before the marker onset. And of course, if you can do something earlier than before, this allows us to get new insights into, for example, the molecular makeup of these cells or the regulations inside those single cells. So that was, that's 5 years back now, right, that we published that. We wondered if we can do the same thing for human cells with clinical relevance, right? So now if you look into human cells, you find many of those on these blood smears here. This is a typical blood smear that you can have in the clinics or in a laboratory. And why do we smear that out? Well, pretty much similar to 180 years back, when Rudolf von Virchow diagnosed leukemia, or defined actually the disease leukemia for the first time by looking under the microscope on a blood drop smeared out from a patient suffering from leukemia. And we do this very similar thing today, every day. So if you don't feel good, one drop of your blood is taken, smeared out on a glass slide, stained and dried, and then inspected by a trained cytologist in the clinics or the laboratory. And here we are, that's a typical microscopy image, right, of a blood smear of a patient or a healthy person. And what you see here, two types of cells, red blood cells, tiny ones, and white blood cells, the bigger ones, the pinkish ones, right? And now what cytologists in the lab do now every day is basically counting hundreds of those cells for each patient and figuring out what cell types you have in there and what the frequency of those cell types is. So, for example, you see that these white blood cells look pretty different, right? Some have a roundish nucleus, some have a granules as nuclei. So they are pretty different. And if you're a trained cytologist, you're able to discriminate actually 30 different of those white blood cell types. And you have to do that because depending on the relative frequency of those, you would either say that the patient is healthy or the patient suffers from some severe blood disease. Okay, now this, right, is a prototypical example for a deep neural network being applied on something, on an image, because that's what those networks are supposed to do, actually recognizing and classifying objects, right? That's why Facebook and Amazon have developed them, and that's what we can do, right? So we would like to have an algorithm that it detects the white blood cells here and puts a label on them so that we can help the clinician or the cytologist to do the job. Okay, so the question for us was now, can we take the model that actually we developed for the mouse blood cells, can we take that and use it to classify blood cells with AI? We looked around for data sets 5 years back and didn't find any. And that is because people, I mean, they look on a microscope, but of course they don't do the extra work, right? And digitize the smear. Why should they, right? They look at the microscope and that's it. So actually there was no digitized data set out there. So we teamed up with Carsten Spiekermann, a hematologist here in Munich at the LMU in the clinics. And he went into his archive and selected the blood smears from 100 patients suffering from acute myeloid leukemia, a pretty aggressive leukemia subtype, and 100 patients which did not suffer from AML, but some other disease. And then what we did was basically taking those, digitizing them, and actually digitizing single cells in each of these blood smears. And there was a tedious work with a microscope here from a vendor close to Munich, Pritzi Point, which was the only vendor actually that allowed us to use oil immersion and to get up to a really high magnification, where Carsten Spiekermann said this was sufficient and actually of a quality that would suffice also in his clinics to do a diagnosis. So we did the same story basically again, right? We tried to identify acute myeloid leukemia blasts. So these are the cells which only appear in the blood of an AML patient, and then basically trained a network with it. So supervised training, I guess you know that, right? For supervised training, you need an image and a label. So in order to train a network here, right, we had to come up with labels, meaning we had to talk to the experts again and ask them to annotate, well, 18,000 leukocytes. So they had to look at 18,000 single white blood cells. In order to do that efficiently, we wrote a little online web interface where they could do that at home or at work whenever they had time. So after a couple of months, that work was done and we could train our network. And the task for the network was to discriminate blast cells from benign leukocytes. And I guess the audience here knows roughly how that is done, right? Conceptually, you show such an image to a deep neural network and you say, this is a blast cell. You show the image plus the label. And then you show an image of another blast cell to the network and say, this is a blast cell. And then you take a benign leukocyte and you show that to the network and say, this is now a benign leukocyte. And you do that again and again and again. And the network is asked to extract the most meaningful features from these images that allow the network to basically do the classification, right? To discriminate blood cells from benign leukocytes. So also, I guess you know that, right? You do that with a trained set. You keep a test set apart, and then on the test set, you evaluate how well the prediction works by taking an image from the test set, letting the algorithm predict what it is, and then checking if the predicted label matches the label in the ground truth from basically the human annotator. If you do that, you can calculate specificity and sensitivity. These are two different performance metrics. And you see that our neural network here, this is the line up there, this line you get if you change the threshold of the final prediction. And you see that basically we're very, very good, right? We get high specificities, high sensitivities. We get a accuracy of 99.9%, which is great. But of course, in the end, whenever you have this AI in a medical setting, you want to know if this is better than the human, right? So you have to compare it to the gold standard, with which is the human. So we showed the test set to another human expert and asked her to do the same labeling as the network did. And what you see here is that actually our classification accuracy is comparable, if not a bit better than the human expert. And of course, once that is running, you get a speed up, right? If the algorithm is implemented, this can be done in seconds and of course, anytime. We also published the code, and we published also the data in the Cancer Imaging Archive. So if you want to take a look there, 18,000 annotated leukocytes with the labels, you can play with it and retrain your own algorithm and figure out if you maybe can beat our performance here. Good. Now, if you talk to hematologists, they say, well, blood smears are nice, but the real severe diseases you want to figure out if you use bone marrow smears. So what's a bone marrow smear? Now, you can take a drop of blood, right, from any one of us and smear it out. To get to the bone marrow, actually, you have to punch into a bone, and that is relatively painful, but you would do that in order to figure out if you really suffer from a severe blood disease like AML or not. So when this is done, right, doctors punch into the bone, they suck out a bit of your bone marrow, and then this bone marrow is smeared out again on a glass slide, and again, dried and stained. Now, bone marrow is not a liquid like blood, but it has more cells in it. They're more dense. You have more different cell types in there because also the immature cells, which shouldn't end up in the blood, are there. And you might have some bone remnants in there, right, making the whole thing more messy. And also, so you have more cells, have more overlap between the cells, and you have, it's hard to scan because actually the focus level of the smears changes. So instead of, I don't know, doing maybe a whole slide scan into minutes, that would take here probably an hour or so. Still, people did that. And we were lucky enough to being able to collaborate with the Munich Leukemia Laboratory here in Munich, one of the largest laboratories in the world to do leukemia diagnostics, and with colleagues from the Fraunhofer Institute here in Erlangen, who developed a microscope specifically for that. Together we scanned 961 patients. We got 170,000 single cell images, and those basically could be compared, or could be basically put into 21 different cell types. And here you see the different cell types, and you see that they are already quite different, right? Again, they all stem from the same stem cell, but during the differentiation steps, they turn out to look very different, and they turn out to have, of course, very different functions, right? The one attacks a pathogen, the other releases antibodies, for example, etc., etc. So same game here, right? Can we discriminate those from the others? And here you see basically a matrix, which is going to be a confusion matrix. I'm going to explain it in a second a bit better. The more diagonal that matrix is, the better it is. So here we can also do that pretty nicely, apart from a couple of cell types where we have a couple of confusions. If you talk to experts, they say, metamonocytes and monocytes, that's actually pretty hard to discriminate. And depending on the expert you ask, you might get different outcomes. So there's some label noise for sure in that. And also then you see here that for those types down there, we have a couple of confusions. And if you look at the number of cells, this turns out to be clear, right? Abnormal eosinophils, for example, we only had a few. I mean, you could count them on one hand, I guess, right? So whenever you have so few cells, of course, you cannot train a proper deep learning network. So that's where the approach necessarily fails. Look into a paper, we published that last year in Blood to get more details on the bone marrow aspect. Okay, interestingly, whenever you do clinical AI, you are asked if you understand what you do, right? If you can explain what you get. And why is that? Well, if it's about what to buy next on Amazon, then you might tolerate errors. If it's about starting a chemotherapy, yes or no, you don't want to have errors, right? So you want to be pretty sure that what your algorithm predicts is sensible and robust, right? So how can we explain what we get? Well, one way to explain that, and of course this all runs under the label of explainable AI, which you might have heard of, is, for example, to take these images and then figure out which of those pixels in those images are most interesting for the final prediction. Here you see a smooth grad analysis. It's one of a couple of possible analysis you can do with these images. And you see that actually the higher or the whiter the pixel here, the more important are the pixel for the final classification. And what you see is, well, at least the pixel inside the white blood cells seem to be the most important cells, pixel, right? Which is good, because otherwise, if we would learn something from the background, then this would be an indication that something is wrong with our algorithm. And this at least tells us that, yeah, they're going to use the same pixel that a human would probably do. Okay, so we focus on the random image regions here. That's good. Now, next step here in these analyses would be that, of course, at the end, we don't want to classify blood cells, but we want to classify blood diseases, right? We want to figure out what a patient suffers from, and we would like to assist a clinician with the decision making. And you can imagine that we get more and more, since we become more and more people, people get older, so diseases get more prevalent. At the same time, we get less and less experts in the pathology labs or in the cytologist labs. So we need this kind of support, and we want to figure out if we can classify blood diseases and not just blood cells. And for that, we looked into a different cohort where we now have roughly 100 of people suffering from, or between 1,500 people suffering from different AML subtypes. Actually, acute myeloid leukemia comes in different flavors, basically depending on the genetic makeup. So if you have a mutation in the gene PML-RARA, for example, then you have a different AML than if you have a mutation in the gene NPM1. And that is relevant because it also affects the possible treatment you might get. Now, we'd like to discriminate those subtypes, and we have stem cell donors as a control type. And let's look into one of those people here. We have 400 roughly scans of white blood cells for each of these patients. And if you now zoom into one of those cells here, this is a particular cell from a patient having a mutation from PML-RARA, and that actually turns into a disease which is called acute myeloid leukemia, APL, acute promyelocytic leukemia. And now if you look at that cell, you see that, oh, there are these strange structures, right? It's not just a nucleus and a cytosol, but there are these rods, and they're called our rods. And interestingly, those are diagnostic for that AML subtype. So whenever you see those our rods, you know that this patient suffers from AML, PML-RARA. It's not the other way around. Not always that then you have the disease that you want to find these our rods, also because they are relatively rare, right? So if you are lucky and find such a cell on the microscope or in the scan here, then you know this is PML-RARA, but you can deduce it from the way around. So our rods are diagnostic. Interesting. So let's see if we can now discriminate the disease. Unfortunately, in this case, having hundreds of thousands of single cell images, we do not have annotations, simply because it's too expensive to ask anybody of the experts to do the classification, to do the annotation. Though we know that all of those cells come from a patient that suffers from AML PML-RARA. So we have a bag label in the parlance of machine learning, but we don't have an instance label. Luckily, there's a solution for that, and this is called multiple instance learning, meaning instead of showing instances to the machine learning algorithm saying this is a blast cell, this is a granulocyte, this is a monocyte, now you show a bag of cells and saying, well, whatever's in there, this comes from a patient suffering from AML PML-RARA. Take another bag. This comes from a stem cell donor, etc., etc. And you hope that the algorithm is, one, able to extract the relevant features from the instances, and two, able to at the end come up with a final diagnosis. Ariel Sadafi in my lab published that in MICA in 2020. Actually, this is the algorithm. So you take such bag and you put it into multiple instance learning. And now what Ariel did, he added an attention label to that. Actually, co-books did that, or colleagues did before that also, but Ariel applied it as the first one to blood cell images. And what you get then is basically a prediction for each of the 5 subtypes. And we did that here. So we trained a machine learning model with a training set, and again, figured out how that worked in the test set. And now here, again, you see confusion matrix, and let me explain it now what you get here in detail. So on the y-axis, you have the ground truth. Actually, that's the label from the expert. In that case, actually, it's the label from the genetic analysis. Now you might wonder, why do I bother with images when there's a genetic analysis? Well, genetic analysis is expensive. It takes much longer than looking under the microscope or taking the scan. And of course, you need a lab that is able to do that. So if we can speed up things with deep learning algorithm that runs on the scans, and if we can do that in, let's say, low resource settings, then of course we can, for example, detect something like AML P-rara quicker, and we can detect it all around the world whenever. And of course, these are a couple of things that we would like to do. So let's have a look at that. On the x-axis, we have the prediction. So in that case here, all of the 60 stem cell donors are also predicted correctly as stem cell donors. That's good, right? Because otherwise you would predict that a healthy person is actually suffering from leukemia. Now, what about the others? AML, PML-RARA, 18 of the 21 patients are also predicted correctly as that subtype, while we miss 3 of them, but I say we put them into the wrong bags, right? And the thing looks similar, maybe a bit worse for the other 3 subtypes. But remember, it's not clear actually if there is a clear morphological correlation between the genetics of those diseases and how the cells look. I mean, for PML-RARA, it is, right? I showed you the other words. For the others, there is some indication. NPM1, you might have cells that have a particular nuclear shape. For the others, it's not clear. So the fact that we can discriminate those genetic subtypes here is already pretty good. And we can go next and now again look into explainable AI and focus on the question, which cells are most important? Actually, if you look into the algorithm of ARIO, you're going to find that we have these attentions on the bottom here. So we learn not only to predict the final class of the bag, but we also learn an attention value, basically a scalar, which tells us which of the instances is contributing to what extent to the final prediction. And that is great because then we can look into that and figure out which cells have been important and then go back and ask which they are. So that's exactly what we do. We're sorting cells into low and high attention and then checking if what we get is making sense. Here's one patient where we sorted all the single cells according to the single cell attention. Low attention cells on the left are deemed not important by the algorithm. High attention cells on the right are deemed very important by the algorithm. Now, we went back to our colleagues and asked them to annotate the cells of that patient, please, which they did. And basically, we asked them to categorize the cells into something like relevant for AML, suspicious for AML, not suspicious for AML. And then, of course, there were cells where they said, I don't know what that is, really. So this is what you get here. The black dots are those cells which have been deemed suspicious by the human expert. The gray ones are not unsuspicious, and the white ones have no clinical assessment, basically. And if you look at it, you see that nicely those cells which have the highest attention from our algorithm are the black ones, which are also deemed suspicious from the expert. So meaning our algorithm looks nicely enough at the right cells. And if you now check into these single cell images, the experts might say, oh yeah, indeed, this looks like a blast. So that, I guess, I would have taken as an AML patient. Good. Now, we trained this algorithm with the bags. We can also use it now to basically take a single cell, like the one here, again with arrow rods, and then push it through our model, which we called Similia. And what you get is a single prediction for a single image. And if you do that, you can basically look at one patient. Let's call this patient here JTF. And then each stripe is one single cell. And then the color tells you how the network thinks that that cell here belongs to, so into which of the 5 different classes, and that you can do for all of the patients. Here you see the PML-RARA cases on the left-hand side. You see that for the correctly predicted PML-RARA patients on the top, most of the cells are also predicted as PML-RARA. On the right-hand side for the controls, most of the cells are deemed healthy control cells, although you sometimes find cells that are also deemed a bit diseased or a bit AML suspicious. So what else can you do? You can drag out the cells from all the patients which have the highest attention for that particular cell type, and this allows you now to basically come up with a nice idea of that disease in terms of single cells. So, for example, Marrow, you see that the cytosol is pretty granular, I would say. And for the control cells, these are healthy-looking granulocytes. So, again, looking into the high attention cells here shows us that basically the algorithm is doing the right thing, and we're getting cells out that people would look at or deem suspicious for the AML disease. Now, how many cells do we actually need to make this kind of assessment? Interestingly, the WHO says that in the lab, people should look at 400 of those cells to make a final assessment. Let's see how many cells our algorithm needs. So what did we do? We basically sampled for each of the patient images, basically subsamples of images of different size. So let's say 1, 2, 5, 10, 20, 50, 100, and then predicted with our trained algorithm how well we would be able to identify the probability for the ground truth class for those different subsamples. And we did that for all of the diseases. Here you see the result for RUNX 1T1, which is one of the 4 AML classes, and the gray lines are the different patients, and you see that for some of the patients actually probability is low, so these are going to be our misclassifications. For some of the patients, we very, very quickly reach a high probability, and on average, we see that from roughly 50 samples on, basically, our algorithm is able to predict very robustly the final ground truth class. Good. This is about blood and blood smears, right? Of course, there are the tumors around, as you will know, right? Many solid tumors. And if you talk about blood diseases, of course, you would look into lymphomas, which are basically also diseases of the blood-producing system. So you have basically solid cancer in the lymph nodes, and here you see one of these images. Obviously, you might know these histological sections here. It's a different type of image, so it's a different type of material. Actually, this is now tissue, which is dried and then cut into 3 micron thick slices and then stained and put on a glass slide and then scanned. And you see that, of course, here it's not just the cells you would look at, but you would also look at the context of the cells and the tissue architecture maybe. So we wondered if we can discriminate lymphoma subtypes with a similar approach. And again, we don't know exactly which lymphoma subtypes are useful to look at, right, or difficult to discriminate. So we teamed up with Rico Weichert and Christina Schwambaum at the pathology lab of the Klinikum Rechts der Isar, again here in Munich, and we assembled a cohort of 4 different lymphoma subtypes and a control class, and we basically did the same thing, right? So we trained, again, a multiple instance learning algorithm because actually these images are huge, right? They are on the size of a gigabyte, maybe even larger typically, and in order to deal with them, you have to cut them into small patches, and once you do that, you can only have a bag label, but no instance label. So let me know if the noise in the background is too loud, then I close the window. Okay. Multiple instance learning on these patches here, you do again the very same thing. Here, a nice work by Daniel, a student in our lab who used also again the attention multiple instance learning, but also a transformer multiple instance learning, a relatively new machine learning approach, which gives you attention between the patches. And here you see the confusion matrix, which looks again pretty good, and pathologists are intrigued by the fact that we can discriminate those 4 lymphoma subtypes, which seem to be difficult to discriminate by eye. Tiffany, is it too loud or is it okay?
It's fine, I don't hear. That's fine.
Very good. That's me.
Oh, now I can hear a little bit, but it's not a problem.
Okay, very good. Good. Now, question, does this also work for data from the UK? Right, so we trained a model, it works for data from Munich, good. Now, can people all around the world benefit from that? And I would like to refer to work from Thomas Frooks at that time at MSK New York, where they looked again at computational pathology using weakly supervised learning, same approach we used here. And what they did is now, since MSK is basically a clinic that not only has its own patient samples, but also gets patient samples from external sources from all over the US in order to make an assessment. They had not just their slides, but they also had external slides, meaning slides that were stained a bit differently, or maybe cut a bit differently, or handled a bit differently. So they now trained a network to figure out how well they could do that. And fun fact, if you look at how large that data is, again, one of those slides is typically beyond 1 gigabyte. The size of that data here, right, is multiple sizes of the ImageNet database, which is used for training machine learning algorithms on natural images. And what they find in the paper is that if you have a very heterogeneous source of images, physiological images, right, you need at least 1,000 images, or maybe even more, 10,000 images, to get a machine learning algorithm that is robust with respect to the source, right, which is able to basically hopefully focus on the feature, but not on the source, to come up with a final assessment and to predict a disease or sub-disease. Okay, well, one of the tasks in that field is obviously to bridge that from proof of concept to production, right? At the end, we don't want to just play with it. We want to have patients and clinicians benefiting from that. And Thomas Fuchs, which I just showed you, he founded a company, Page AI, a couple of years back, and basically Page now received a year ago the first ever FDA approval for digital pathology product. This is for prostate classification. So actually you can now, once you have digitized your slides, go ahead and use that for classifying your slides. Interestingly enough, it takes a while to get there, right? Although there are so many different methods now out and also some data sets out, it's actually not that easy to take algorithms and basically bring them into production. I mean, there are a couple of regulatory aspects as well, but alone from the algorithmic perspective, it's pretty hard to basically redo what other people did. And if you're interested in that problem, have a look at our Nature Medicine review, where we reviewed that topic and basically asked for making deep learning algorithms in computational pathology more reproducible and reusable by publishing code and saying exactly what you do with your model. So have a look at that. Good, final point here. Can we go superhuman? Well, what do I mean by that? Can we detect diseases where the expert fails, actually? You know, one, we have the thing running, we can check for that. And actually that's a fun thing to do, because there are some diseases which experts say are impossible to detect from the blood smear alone. So let's go back to blood smears and look at a disease which is called myelodysplastic syndrome. If you talk to the experts, they say, no, we cannot identify that disease from a blood smear. It's a pre-form of AML. So having, again, access to a very nice data set from the Munich Leukemia Laboratory, we figured if we can train an algorithm that is able to detect myelodysplastic syndrome from reactive or healthy samples. That is work by Peter Lienemann, actually an MD, basically a medical student that is doing his doctoral thesis with us. And what Peter did now was training an algorithm and then figuring out what the most important cells are. Actually, we find our accuracy in detecting MDS, myelodysplastic syndrome, is above 70%, so much better than random. So there is a signal in there. But if you look at the cells now that the algorithm picks, you find basically a large heterogeneity, right? So there's not a single cell type, for example, or let's say a single pattern in the cells that would allow us to say, that's what the algorithm is using. No, we get here, as you see, one of those lines here is one patient. We get a mix of cells and cell types, which leaves us a bit baffled, and we basically go back to start, right, and have to figure out, why is that? We want to understand why that is. So we have to employ, again, explainable machine learning methods. And one of those methods which you can do is basically going back to the feature-based approach, basically not using a convolutional network or deep neural network at all, but doing it as like 15 or 20 years ago, basically taking the single cells, segmenting them, and then extracting features like size of the cell, roundness of the cell, color of the cell, etc., etc. And that we do in order to be able to look into the features which are most important for the final classification. And that is what we're looking at the moment, because we'd like to understand why we're able to do that. Oh, right, here's the performance actually of that. And yeah, as I said, it's much better than random. So there is something in there. The challenges of this AI for hematopathology approaches, I guess, is threefold. So one is, how do we ensure explainability on all levels? That's what I just told you, right? We want to figure out why we're able to do that. And we want to figure out it in terms of the instances, but we would also like to figure out it on level of the pixel. So we're trying to get these, like the smooth grid I show you, these type of methods into our multiple instance learning approach in order to be able to figure out if the algorithm, for example, picks up the hour rod, which I showed at the beginning, and also uses that kind of structure for the final prediction. Can we construct robust models? Again, this is the issue of if you have a model, right, and it's trained on data from Munich, are you able to predict data from the UK with that? That seems to be more complicated than we thought, actually, and a couple of people are working on that. Because as a human, if you saw a couple of different data sets, I guess you are able to figure out, basically to discriminate or entangle the features from the domain-specific features like color, size, scan, resolution. For the model, that is much harder, it turns out. So actually, we are looking into a couple of domain generalization model methods at the moment to figure out how we can do that, coming up with a robust model in the first place. And then finally, of course, how can we bring models from proof to production level? And I guess it's great that we can collaborate with the clinics in Munich and also with the Munich Leukemia Laboratory, which are very open to collaborate with us and to figure out how we can design these models, how we can bring them into the pipeline, and also evaluate if actually, or how much actually we gain in efficiency when we use that for the daily job of blood smear or bone marrow smear or histopathological slide analysis. This is the lab, and maybe just let me mention a few people. I don't know if you see my cursor, you see, I guess, right. This is Rao, he's working on robustness issues. This is Salome, who just published in MICAI a way that we identify outliers, basically, both on the single cell side, but also on the patient side. Rahele worked on methods that would be able to, identify single cells in the blood smears of different sources and make the whole algorithm more robust. Ali is also working on domain generalization methods, and Ario we have here, he basically brought the multiple instance learning expertise into the group, and we are extending again on these aspects to make our models more robust and more performant. The other people work also on super interesting projects, which I didn't talk about today. Happy to do that maybe in another round. And of course, as I said, there are these statistical models like the deep learning neighbors, and there are the mechanistic models. And basically, in my lab, we try to basically also now get them closer together, right? We're figuring out how we can, for example, we have the knowledge we have about a system and use that in a deep neural network, or the other way around, figuring out what features, for example, are used in the neural network and which features we know of they might correspond to. Thanks to our collaboration partners. I mean, without the data from the labs and the knowledge of the clinicians, we couldn't do that work, right? The communication between the computer scientists or the biomedical people and the clinical experts, that's key to all of the projects I showed you. So thanks to the MLL, Carsten, and our pathology partners. Again, we try to bridge these two things, and this is what we currently work on. If you're interested in that, write me a mail or look into our grad schools. Actually, this is a bit of a larger picture. So we have a couple of health AI groups working at Helmholtz Munich, different aspects of that, different aspects of the level of translation. And there's one thing which might be appealing to you. So we just opened another call for EPI Cross Borders. This is an international grad school between Helmholtz Munich and the University of Edinburgh, where we basically have open positions for PhDs to be done between the two sites. So you will be shuttling a bit between Edinburgh and Munich and work between two groups, one in Munich, one in Edinburgh. And if you're interested in that, check out the EPI Cross Borders website or write me a mail if you don't find it. Deadline is November 12th, and we have a couple of interesting projects, and we would be happy to get interested students to apply for that program. Wonderful. Thanks for the co-workers, and thanks for the students, and thanks for the funding. This is how Munich looks from a bit above, and I'm looking forward to your questions.
Wonderful. Thank you very much. That was very exciting. Were you kind of interested as well in kind of setting up a company for AI diagnostics, as quite a few people have done? I mean, we have Marcel Gerung, who has a company called Cytologists, which is a data-driven diagnostics company, which came out of Cancer Research UK. I'm just curious, but I think you showed all your work was kind of academic research with clinicians, so translational, but it wasn't going commercial, I think.
True. Yeah. I mean,
that's my question.
Very important. Computational pathology is, of course, one of the fields where a lot of is happening, right? A lot of startups are popping up, and people trying to, design not just algorithms, but really solutions for the clinics.
Yeah, it's very interesting.
Did I lose you? No, I haven't lost you. I didn't do that. Try that myself. Actually, Stephanie. At the moment, I try to push things forward, and we have the Munich Leukemia Laboratory as an excellent partner who are interested in, getting that done in their pipeline, basically. So I'm totally happy to work with them and try to push things as much forward as possible with them. Yeah, no, it's kind of just kind of just curious. I saw you had like Moonlight, I think is one company. I saw it on your LinkedIn page, something you kind of liked. So I'm kind of curious. Well, we're going to have the quiz now. So we always, after the talk, give the speaker a few minutes to recover. And we do the quiz, which you had sent. And Nick has actually prepared the used the quiz question, and hopefully some people who are going to participate. Come on, everybody who is there, could you please sign in? And there's the link, the Mentimeter one in the chat.
Can you see the quiz in the?
Yes, I see it, Nick. I'm very much looking forward to that.
We've only got one participant so far, so we need a few more people to sign in. If you look in the chat, the link is there.
Maybe just put it in again. Some people may not have seen it.
I don't know. Where are we?
I'll do it. I'll post it now. Yeah.
Got it? Because I can't get. Yeah, I can't.
Yeah.
There we are. There we are.
Yeah, that's it. It's just a good reflection on actually what you have shown us and how we can.
Yeah, very.
Quite a good summary, and usually questions come up which come up anyway. So
yeah, let's see if we get more than 2 people on that one, because we've got some good questions this week. Big prize, the Foldscope will be winging its way to the lucky winner when.
Oh, that's a good point. Actually, I guess I found that Foldscope on your website, and I looked into it, and it's is it as nice as it looks? You had them in hands, right?
Well, it's a very educational thing, so it's based I think it's almost a bit like the first microscope ever by Leeuwenhoek, because it's something, it's not easy to use, but I think it kind of demonstrates really nicely all the kind of steps and developments. And obviously it was one of those jokes in the beginning when everybody was sitting at home, how can we get access to microscopes? And this was the easiest because it goes through the letterbox and it's small and foldable. And
I think it's a great prize.
And it's a lovely, I mean, for kids, and you can, put it together, and you can learn something even if you're a grown-up microscopist, I think.
Well,
Get a few people signing up. What's happening?
We've got plenty of people in the audience, so we should get a few more participants. Come on,
don't be shy. Go for it.
There.
I also invited some people from my group to join, so also you guys get for the Foldscope.
Exactly.
Come on, everyone.
Yeah, because actually it's you have to be quick on the buzzer, as it were. So if you're fast, you'll get the prize. It's not just knowing the answer; you've got to be quickest as well. Oh, here we go. We've got a few participants. Right, let's just go through. Let's start the quiz. Oh, yes. All right.
Now we get, I think it was the thread. I think you're Carsten,
Explainable AI.
Air in your lab.
That's what we need, apparently, or both. Okay. So here we go. Question 1. Come on. All right. Answer fast. So it's all about being quick. What's the size of a typical digitized histopathological slide? Is it 1 kilobyte, 1 megabyte, or 1 gigabyte?
Oh, that's interesting because
Let's see what we've got in the way of answers. This should be an easy one to kick off with. People got 1 gigabyte, so we've got 6 people in the running so far. Let's have a look at the timings there, because we'll see who's actually was quickest.
They are pretty large, right? I mean, that's what what giving us headaches, right? Dealing with those huge slides has a couple of mythological.
Yeah. So Dragonite is faster, but there's 1, 2, 3, 4, 5 people on your tail. So let's see what happens next. Okay. Next question. How many human cells do we consist of? Is it 30 billion, 30 trillion, or 30 quadrillion? And the answer is... Dun, dun, dun.
I made this more complicated in my talk, I guess, that it would be, right? So on purpose, obviously.
30 trillion was the answer.
Sorry if I said it wrongly.
well, it was helpful to remember. And
I double checked, actually, according to the...
Thank you, Nick. Great.
The internet said 30 trillion. Okay, let's see how we're doing on scores. Here's the leaderboard. Oh, Dragonite, I think you're still in the lead. Puffy and Elmo on the tail. Okay, right. Still room to catch up here. Question 3. Which weakly supervised approach did we use? That is Carsten. Was it multiple instance learning, unsupervised domain adaptation, or Snorkel, an open source system for quickly assembling imaging training data? And the correct answer is dun, 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 multiple instance learning.
Nice. People seem to have listened.
I liked Snorkel though.
Yeah.
I was sure nobody fell for that one.
Okay. Let's see where we are on the leaderboard now. 7 people got that one right. Okay. Ooh, other people are catching up. Dragonite is still a commanding lead. Come on, we need a few more quick answers from a few people here. Okay. Question 4. How many cells seem to be needed for proper AML subclassification? I've definitely heard the answer on this one. Was it 500 cells, 50 cells, or 5,000 cells? If you were quick here, you might have pushed yourself in the lead. Everyone's voted. Oh, it is 50 cells. Well, this might have changed things. Let's see how the leaderboard looks now.
Interesting, right? I mean, WHO recommends 400, and we in our algorithm needs only 50. So
Puffy. Ooh. Okay. Puffy and ET are on your tail, Dragonite. So, one more question to settle this. Okay, last question. Answer fast to get more points. Which algorithm allows you to estimate the importance of single cells for final classification? Is it hazy research learning, active learning, or attention-based multiple instance learning? And the answer is attention-based multiple instance learning. All right, everyone got that one. Okay, let's see who the winner is.
Yes, probably.
Dragonite is probably there.
Wonder if Dragonite is from your lab now.
Oh, yeah, I think that's fast enough to hold on.
exciting.
Okay, Dragonite, well done. If you could just make yourself known to moderators in the chat, that's fantastic. Foldscope, we're winging it away.
Hi everyone. So Dragonite, that's me, but that might be a bit unfair because I'm a postdoc with Carsten.
Fair
Yeah,
enough.
but I mean, you haven't heard that talk that often, Max. So I'm glad to see it.
No
Often enough,
problem.
maybe.
Well explained, I would say.
Well, Carsten wanted to see a foldscope as well, so I think the deal is you have to show it and build one up yourself or together, so you can have a play.
Very good. Yeah,
Congratulations.
I try them for sure.
Nice.
Very good.
Wonderful. And we had some other questions from the audience. So, I mean, we give some space here. Anyone who wants to maybe kind of unmute and make themselves visible and ask the question. I think there was one. I think there's someone, Nero Hayes, she's asking a few questions. Like she's asking, do your models generalize well on independent test sets? I guess you answered it a little bit, but I mean, independent test sets, I guess, is more than spoken about, in which you probably see the answer, the question here. So what techniques have you tried?
Yeah, you can go to the chat, right? Independent test set. Yeah, I mean, this depends on how you define the test set, right? The easiest would be just to assemble it randomly, right, from the whole data. If you have now patients, of course, you should not take the test set with single cells, but of course you should stick to the patients, right? So that's what we did. We basically left out whole patients and then tried on that, and that was how we assembled the test set. That is good. It's not an independent test set because it only, it comes from the same lab, same scanner, same approach, basically. So we do not yet know how this works on an data coming from a different site. And actually, it's also not that easy to say because, as I said, there is no other publicly available data out there for these kind of questions, or it's coming at the moment, right? So it's exactly what we try to also to bring people to make them publish the data so we can train models on that. And the one we trained so far with the 4 AML subtypes, I mean, there is no comparable data out there yet. So let's see how that works if somebody publishes data. What techniques have you tried for dealing with issues like overfitting and model translation? So again, you split up your data, right, into training, validation, and testing set. And with that, you can basically evaluate how good that is on the test set. Actually, what we did was using a cross validation approach. So we do it multiple times and basically average over the results we get at the end. Model generalization, I discussed that, right? This is something we try at the moment. So we assemble data sets, we try to assemble data sets from different sources and also figure out what algorithms are there that allow us to generate more robust models, be it augmentation or be it figuring out what features actually belong to the source and which features belong to the class. And then I go to Sotiris question, Stephanie, at the end.
Sorry, I think these were the questions which
Sotiris.
Sorry, yeah.
Sotiris is next, right?
Oh, yeah, that's right. Yeah.
Synthetic data. Are you planning to merge the different types of models to get optimal results? Yeah, synthetic data. So for the cells, no, I haven't seen synthetic data. What we try, I think. No, you need patient data, I guess. If you want to classify AML, you need patient data. What we tried to do was, though, to synthesize patients, basically. We tried to basically assemble cells from different patients and basically enlarge the number of data sets we have. Or there was a recent publication where people talk about pseudo bags, basically splitting bags up into smaller, splitting patients up into smaller or patients with smaller bags, which you can do to train. These are things you can do, I guess, to increase the efficiency. And we played with it a bit, but have no real reportable outcome for that yet, if it's increasing or accuracy or not. Are you planning to merge different types of models to get optimal results? Well, again, I guess it's the generalization question, right? Ideally, we would like to, whenever we tried that, actually, if you merge data sets or if you merge models from different data sets, typically your performance deteriorates on the one data set, right? I mean, on all data, it gets better, but on the one you look at, it deteriorates. And then again, it's a question, if you want to do that, right? Ideally, we want to have something that would be applicable to any type of scans you get, so that you also can apply it into, let's say, low resource setting. For somebody like the MLL, a research laboratory, right? They don't have an interest in a generalizable model, right? They just need a model that works perfectly for their setup. So I guess it also depends on where and how you want to employ the model to basically do that, right? Generalizing, but then at the cost of getting a bit lower accuracy on the one data set you might be interested in.
How much difference does it make in terms of the imaging systems you're using? Because I presume in the clinic you use relatively basic system, and you don't need like a super resolution scanning system or something for the work. I mean, would it improve, or have you?
Yeah.
I mean, I don't know. It's maybe a bit naive.
No, I mean, there are like, let's say, between 5 to 10 different scanning companies out there, and the scanners are between 20x and 40x. You know, the oil immersion thing, which we did, is not yet commercialized. Also, there is no solution where you put the drop of oil and then do the scanning, because it's also mechanics is pretty intricate here. So it makes a bit of a difference for sure, and also the lighting makes a difference, and the resolution makes a difference. So whenever we looked at that, actually, as I said, right, the model has real problems to be able to generalize from a model that has been trained on one data set to the other, but actually that doesn't work. So either you have to train with data from all different sources, or you have to make an extra effort to apply domain generalization, and that's what we currently try out.
Okay, I have a couple of questions. So how big is the actual gap between proof of concept and clinical diagnostic use? How difficult is it to get across that gap?
Yeah. So we actually wrote another ERC grant, it's a proof of concept grant, where I want to do exactly that, take the algorithm to the Munich Leukemia Laboratory and run it in parallel to the standard approach, right? And then see how big of an improvement you get. There, it is not such a big step, because basically they have computers, they work with it already, they have some software. Now to implement the disease prediction there would be possible, as long as, you know, the hardware manufacturer and software manufacturer is with us, which they are. So I guess there maybe 2, 3, 4 months of work, and then we could run that and evaluate how much time and accuracy can actually, how much we can improve on that. Now, then, to get that standardly working in a clinics, I guess there are a couple of regulatory affairs we would have to deal with, and that's something we haven't done yet. So we first want to evaluate how good we are in terms of timing and accuracy.
So to carry on that sort of vein, if you got an AI network into use for clinically approved use, is it allowed to keep on learning once it's there, or does it have to be set in stone at that point?
Very good question, Nick. I guess for now, it should be set in stone. I mean, I don't know exactly how that is for Page AI, but for now, I guess if you have something approved, you're not allowed to change it, right? Otherwise, you have to reapprove it. So I guess the way Page AI does it is they train the model once with the data that they had, and now they sell that, right? As soon as you keep training, you need something new. I just had a talk with a lawyer in Germany, a German lawyer, last Friday. There is so actually, I guess the government is realizing that, and in Germany they come up with something like, which is called a KI-Verordnung, so a small add-on to the laws we have, where people try to deal with these kind of issues. So let's see what the updates are there, but that's still in negotiation, so it's not yet clear what's coming out of that.
Right, thank you very much.
Interesting. Thanks very much. In the interest of time, I think we need to close now, but also wanted to say thank you for Georgina and Royal Microscopic Society to keep running this and helping us with the administration. We didn't say it in the beginning. It's Georgina or Jessica. They're usually obviously dealing with all the kind of emails, sending out invitations, and keeping with us for the duration of the talk. So. But thank you very much, Carsten. This was excellent and such a wonderful inspiration and overview and what can be done and what you do in particular, obviously with your team at a very high level. It's wonderful to see, and well, you explain it, I mean, so people can learn and actually really benefit from it. So wonderful. Thank you.
Enjoyed it. Thanks for the
Thank you very much.